Buenos dias, good morning. Thank you, Jennifer, and thanks to the organisers. I wish I could say olé and do those amazing dance steps that gentleman was doing last night, but I'd probably fall off the stage and just embarrass myself and you all, so I'll skip the flamenco moves. So, as Jennifer mentioned, we had a wonderful set of talks yesterday and a lot of very beautiful detailed work on the ecology of responses of species to global change and so forth. And that's sort of the theme I'm going to continue with today. On behalf of myself, Rosa Agudo, who's sitting here, currently a postdoc with me and from here originally. And also I'll be presenting work from a number of my colleagues at Museum of Vertebrate Zoology at Berkeley, where I was until very recently. So the theme really is learning from the past to understand the future. It's a very obvious thing to want to do. So the challenge, of course, is that we're moving into uncharted territory. You've all seen these sort of famous hockey stick graphs with whatever's coming dependent on our behaviour as human societies, our emissions, our control of energy usage and habitat disruption and so on. And this graph actually shows two types of challenges. One is just that mean temperatures going forwards are moving outside of anything experienced over the recent past, and in fact the lifespans of many species on the planet now. But the other is we're moving into non-analog climates. We're moving into combinations of temperature, rainfall, seasonality that have no precedence. So we have the phenomenon, as Jack Williams and co put it, of novel climates and of disappearing climates across the planet. So how on earth can we predict what's going to happen to species, communities, ecosystems based on our current knowledge if we're moving into non-analog situations? You just can't extrapolate statistically easily into that sort of framework. Yeah, of course we do it because we have to, because policymakers want guidance on what's going to happen, what should the priorities be, and so forth. And these predictions are really, really depressing. Just a couple of examples here. At the top, some work by Lasorte and Jets on montane birds globally. Red is bad in the second graph. That's just showing the species are going to lose the majority of their range. These are montane species predicted to move up mountains. And in the bottom, some very recent work from Warren et al. Showing the proportion of species predicted on the base of species distribution models to lose at least half of their range under various scenarios. And red is business as usual. Green and blue are meeting emission control targets at different times. So these are pretty serious news if they're at all true. Maybe 50% of plants losing half their range unless we control emissions, down to 30 or 15% if we can control emissions earlier. So the literature is full of this stuff. And everyone recognises the limitations of employing niche models in this way, but we have no other method that is scalable to very large numbers of species. So we're kind of stuck with it for the moment. And of course, the policymakers and managers would like us to be able to predict which species are vulnerable. And this is a really hard thing to do, as anyone who's in the game knows. So if you look at the top left here, there's a sort of a cartoon version of some much more complicated schemes I've seen, trying to sort of go from exposure, sort of mesoscale changes in temperature or precipitation or rates of habitat loss at the top, with the vulnerability at the bottom being mediated by the response of the species and their intrinsic sensitivity and the sort of interplay between those. So we might imagine, as we saw yesterday in some wonderful talks, an evolutionary response, either genetic change or perhaps the role of plasticity in that. And that may work for some species, but we know for long generation species with low reproductive rates, that's going to be a significant challenge, particularly those that are sensitive. So the adapt bit of that response curve looks quite challenging for a large range of species. The buffer bit in that green box, I think, is one that's underappreciated and perhaps understudied. And I've tried to show that with that sort of blue box there on the right. So typically we make our predictions, or we have our predictive surfaces like climate surfaces and so forth at, well, if you go to the GCMs, the global circulation models, 250 kilometres, we scale that down, sometimes egregiously, to 1 kilometre squared. But organisms perceive and operate in the environment, particularly little organisms like I work on, at the scale of a few tens of metres. So so there's this real scale issue in how animals perceive and operate in the environment. And perhaps, microhabitat buffering and habitat choice for thermoregulators or organisms that regulate their hydric conditions is going to be important. And this, of course, is missing from these very large-scale models. I could go on and on about the things that are issues of species distribution models. I use them myself because it's what we have, but clearly we need to improve it, incorporate demography, and so on. So all this is coming down to the issue that even from a conceptual, theoretical point of view, predicting vulnerability of species to global change is extremely challenging if you want to do it at scale of hundreds or thousands of species. So, what are we to do? Well, let's use history. And there's a range of scales of historical information we can play with. So, this is what sort of Rosa and I cooked up. Ooh, how's this work? There you go. We all know that this is a bit we're just looking at, the various rates of future change. But we know that that's following on from a relatively stable Holocene period, but with an upkick in the late 20th century. This, of course, is not a linear scale. It's some weird compressed scale of various things. From the late Pleistocene, these massive oscillations. Earlier in the Pliocene, big oscillations. Going further back in time to the mid-Miocene, there was a mid-Miocene optimum, and so forth. So many species on the planet now have sort of average species durations of 2, 3, 4 million years, depending on taxon. So they've been through a lot of this stuff. Now, of course, there's been extinction as well. From the fossil record, it's actually hard to see pulses of extinction coincident with rapid climate change, except for the mass extinctions, the big five. But otherwise, through all this stuff, what the paleontologists report in the literature, and I'm not a paleontologist, is that you don't see spikes of extinction with these relatively rapid episodes of climate change. Yet all our models looking forward predict what, to all intents and purposes, is a mass extinction. Now, there are some fundamental differences. One is that we're now living on a planet that's massively modified by humans in terms of habitat disruption and consumption of primary productivity and so on. And it may be that the rate of change that's forthcoming is actually higher than even these rapid transitions like the late Pleistocene-Holocene transition. That's debatable. So the fossil record suggests that actually there's a fair bit of resilience in the system. Now, but the fossil record, of course, has limitations in space and taxonomic resolution, representation, and so on. So it gets quite good in some taxa through the Holocene. But then let's think about the 20th century record, which I think also holds a lot of information in what's to come. And that's really what I want to focus most of my talk on. And in particular, the role of collections in natural history museums, herbaria, museums, and so forth, as well as long-term ecological data sets in getting a fine-grained picture of comparative species responses to climate change. So we think of museums traditionally as this sort of stuff, dead chipmunks in a box, lots and lots and lots of them. And I'll be talking a bit about these chipmunks as we go along. But increasingly the use of museum specimens is very multidimensional and often in ways not appreciated by the original collectors of the material. They couldn't have imagined stable isotope analysis. They couldn't have imagined next-gen sequencing of museum skins or fish scales, as Ollie showed us yesterday. So museums traditionally been in the space of specimens, but increasingly now it's merging these with spatial modelling and biodiversity informatics, with environmental data, environmental change in space and time, and increasingly also genomics and various phylogenetic and population genetics tools. So the scope of what we can do with these old specimens now is ever expanding and increasingly relevant. So as one example, I had the opportunity to work at the Museum of Vertebrate Zoology in Berkeley for about a decade, and led this project which we call the Grinnell Project for reasons about to become obvious. But this is the collection. It's about 750,000 specimens, not as big as the Smithsonian or American Museum or whatever, but substantial, very big mammal collection. But what makes this collection special, and many others, is how data-rich every specimen is. So we have not just the specimen, we have the original collecting maps, we have bird census data from the early 20th century, we have the historical photographs, we can go back, reshoot the habitats, and we have hundreds of thousands of pages of field notes, which, as I'll say in a second, are really fundamental to using the information. This is sort of early 20th century informatics. And the museum was set up for this, expressly for this purpose. So these are words from Joseph Grinnell, the founding director, in a paper he wrote in 1910, which was pointing to at least one of the purposes of the museum was to establish this baseline record so people could come back in a century and see what's changed. That's pretty cool from someone in 1910 thinking about what are you going to do with this thing he's building and his colleagues are building. So you didn't have to be a rocket scientist to figure out this was a good thing to do. He said do it. Okay, so we did it. But also, I really like this. There was a comment from an undergrad. I was leading some potential donors, like that worked, around the museum, and we like to connect these rich people with the undergrads who might excite them about giving money to the museum. And one of my undergrads said this. I thought that was so good. A specimen is like a good book. You can read it again and again. Look after specimens. They're great. So what Grinnell did with his team, funded by this amazing woman, Annie Alexander, was to go all over California and collect particularly birds and mammals, document their habitats and so forth. And he was interested, in fact, develop the concept of the ecological niche, published on it in 1917. And that arose from his work, particularly across these transects with very steep environmental gradients. And I'll be talking about this one, the Yosemite transect, but we've now revisited a bunch of these transects. And even then, California was subject to rapid environmental change. This was just after the gold rushes, which had had a huge effect on habitats in the lower slopes of the Sierras. The Sierras is this mountain range running down here. The Central Valley, this is habitat integrity, brown is bad, green is good. You can see the Central Valley there, the agricultural area, already pretty hammered in the early 20th century. But importantly for us, there's been a good deal of climate change, red being increases in temperature and red being decreases in precipitation in the bottom across California, and in a very spatially heterogeneous way. It's not like the whole area has changed synchronously and in the same direction. Some areas have got warmer and wetter, some have got warmer and drier, and so on, depending on local topography and climatology. So this provides an incredible baseline data set of specimens, and again I emphasise these field notes. So from these we could know how many traps they put them out, where they put them out, the daily trapping records, the trapping effort, and because they worked sites over multiple nights, we could apply likelihood or Bayesian occupancy models to the data to control for false absences or variation in detectability. So this early bioinformatics was absolutely crucial to being able to use the collection as we did. So our goal was very simple, and it was the ultimate in hypothesis-free research. We were just doing what Grinnell told us to do. Go out and see what's there now, and document how it's changed, and try and understand why it's changed in the way it has, and then also provide another benchmark. So we're out there collecting all the specimens again, as Grinnell did, particularly for the mammals, so that people can come back again in 25, 50 or 100 years and repeat the exercise. Lots of fun. And a huge number of people involved in this work. I was just one of the ringleaders. Particularly point, Steve Beisinger was a sort of ecological statistician, and Birdo working with us. Jim Patton was really the heart and soul of the enterprise. And also mentioned Morgan Tingley, who generated a lot of the bird data I'll talk about later. So we'll start with the Yosemite transect. And Yosemite is sort of an iconic national park in California. Everyone on the planet's heard of it. It's an amazing landscape. And our transect started down in the Central Valley at just a few tens of meters of elevation up to the crest at about 3,500 meters, and then over into the Great Basin on the other side. So Grinnell and his colleagues worked this area very intensively, and again he was interested in how species replace each other elevationally and with habitat, and along the way developing the idea of ecological niches. And they put in nearly 1,000 sort of man days. Now this sounds very sexist. It actually kind of is. This work was all funded by a woman, but men and women did not work together in the field. She went and did her field work, and the guys went and did their field work, and never the twain shall meet. That's just how it was in those days. Thousands of pages of field notes, specimens, photographs, a very, very rich data set. So this is sort of the nature of the data. So actually, if you don't mind, I'll flip over this side just for a bit of a break. So at the top we've got the grey is the distribution of sampling sites. M is the modern resurvey, H is historic. The black dots are occurrence records for a given species by elevation, from 0 here up to about 3,700 metres. The green ones are shot animals. They used to run around shooting stuff. We can't do that now in Yosemite. We do it all by trapping. But they also provide usable records. And this is one transect up here in Yosemite. We did another transect, the Southern Sierra. And just looking at the raw data, you can see that the lower elevational limit of this particular species has moved up substantially, a few hundred metres, and has done so also in this other transect here. Now, you can take that at face value, or given that we have the trapping effort and nightly records, we can apply occupancy models, which sort of give you the estimated shift in elevation, again elevation here, probability of occupancy on this scale, as a function or controlling for variation in detectability, trapping methods, trapping effort over time, between historical and current and so on. So the data are pretty robust, about as robust as we can get it. So in both cases here again we see this upward shift in this particular species, the alpine chipmunk, that I'll have more to say about later. So that's basically the basic form of the analysis. Now in a sort of more cartoon version, I'll go back this side just to confuse you. What's this one? Oh, the pinion mouse. This one moved up, we're in feet now. They keep using feet in America. It's like chains and cubits and stuff, but nonetheless they do. So this has moved up enormously in elevation. It's actually shifted habitat. So it's called the pinion mouse, and it actually has different subspecies on either side. We showed genetically that the individuals that now turn up right on the Sierra crest in this whitebark pine, sort of montane Sierra pine community, actually came from the east side, and they were your typical pinyon juniper woodland mouse that moved up almost 3,000 feet over 100 years. Just jumped up over the crest. And on this side we see something that's more typical, about a 500 metre upwards translocation of the range over that time. Of more concern than just low elevation species moving up is high elevation species contracting, and this is, I've already shown you one example. The pika one actually subsequently was found not to be as bad as we thought with more surveys. But things like the golden mantle squirrel, again moving up this case about 170 metres in elevation over that time. And of course as they move up they lose area also on the mountain. Because it's real data, not everything's moving up, some things are moving down. And there was some real surprise in the shrews, and this may have to do with the hydrology of the meadows, which has changed. This is a wet meadow specialist, and the meadows are actually now in better condition than they were in Grinnell's days due to habitat restoration. But the basic message is there's a lot of spatial dynamics in this system. Now, we talk about, as one of the major strategies for dealing with particularly global climate change, is protecting big areas across climatic gradients to enable species to move up and down and do all this stuff and persist. And that's pretty much what's happened here. No species have been lost in Yosemite. There were things that disappeared early, the iconic grizzly bear, wolverines, porcupines have disappeared, but that's because of direct hunting pressure and removal in the late 19th century, early 20th century. But in terms of the climate change response, Yosemite has not lost species, it's maintained species richness. It's done what a big protected area across a large climatic gradient should do. And that's good news. More intriguing as a biologist in this question of predicting species vulnerability is sort of the overall patterns. So green here, so again, this is elevation, and each bar is a species, with green representing statistically significant expansions, red statistically significant contractions. So you can see there's a general picture here of some low elevation species moving up. Here's that one that moved down, as I mentioned, the shrew. And then high elevation species contracting upwards, in some cases collapsing in their range, like this one here and this one here. Yet there's a whole bunch of other species that have not changed at all over that 100 years. And in some cases they're closely related. Everything with a T there is Tamias, it's a chipmunk. They're very closely related, and ecologically they do similar things. So this one's a shadow chipmunk that collapsed. This is actually at the southern limit of its range, so it could be a latitudinal shift. This is bushy-tailed wood rat, it's collapsed in its range here. This is the alpine chipmunk I already showed you. But here's a chipmunk, and here's two chipmunks that haven't changed their distribution. So the corollary of this is the local community composition of congeners, of closely related species, is changing. So in Grinnell's time, he paid a great deal of attention, and others subsequently, to how chipmunks replace each other elevationally across the Sierra gradient. That's all changed, and local community composition has changed as species respond idiosyncratically to whatever's going on in this system. And the same is true of Peromyscus. You've got Peromyscus truei expanding dramatically. That's this one here. There's just the west side data. Maniculatus hasn't changed, Boylei hasn't changed, and so on. So closely related species, some respond, some don't. The shrews are doing all sorts of different things, and so on. Now, what's going on? And of course, it's not just one thing. This is a complex landscape over a huge elevational range, but the standout, of course, is warming, particularly of minimum temperatures, nighttime temperatures. Just based on the instrumental record, most conservative estimate is that's increased about 3 degrees Celsius over this 100-year period. So that's, again, the minimum temperatures, not the average temperatures. Maxima haven't shifted that much. And we see signatures of that in contraction of Lyell Glacier. That's the early photo, that's the reshoot. And in meadow invasion by lodgepole pines, growth form of Frumholtz pine, and so on. So there's a lot of signatures there for increasing temperatures. Now, of course, there's also been habitat change. In fact, just in the news last week were massive fires running through Yosemite towards Hetch Hetchy. So it's been a policy for a long time of fire suppression, as there has been in many parts of the world, and that builds up fuel load, so when a fire happens, it's a state-changing fire. It's transformative. And there's particularly been a lot of fire dynamics, so-called seral dynamics, at the lower elevations. But fortunately, we have historical vegetation maps from the early 1930s, roughly matching the historic surveys, and we could remap those in the same language of vegetation in the present and look at does expansion of habitats over time, whether due to fire or temperature or rainfall change, does that predict what's happening to the species? This is Maria Santos's work. And the answer is it sort of does for the expanding species. The green dots here are expanding species, and that's their change in species range, change in their respective habitat ranges, and it sort of works for the low elevation expanding species. It seems to be something to do with habitat expansion, probably related to fire, but maybe climate as well. But it doesn't predict the high elevation contracting species. There's almost no habitat change measurable at high elevations. It's a much more static system. Now, we can ask, is this just a phenomenon at Yosemite, or do we see it replicated across different transects? So Kevin Rowe and others, Karen Rowe, led an effort to, that's the Yosemite transect. We added in a Northern Sierra transect, Southern Sierra transect. Not all species are in all places. And the low elevation expansions were very heterogeneous from one place to another. It wasn't the same species doing the same thing in different places. That sort of fits the idea that maybe this is more to do with local fire regimes or forestry practices at low elevation affecting species ranges. What was consistent is contractions of high elevation species, particularly in upward contraction of their lower limits. We saw this again and again, often in the same species across different transects, despite slight differences in the local pattern of climate change. But again, it's heterogeneous across closely related species. So in some cases, we took this further. This is Belding's ground squirrel, which is very observable. It just sits up, I call it a picket pin. It sits up in its meadow and screeches at you when you walk onto a high Sierran meadow. Very observable, very cute. And so Tony Lynn Morelli did much more extensive surveys of this, and it's disappeared from almost half its sites, half its historical locations, and we've seen no colonisation of formerly vacant meadows based on the historic surveys and resurveys. So no colonisation and a whole lot of local extinction. Interestingly, the exception here was what she called anthropogenic refugia. Things like campgrounds, or in one case, down on this side near Mono Basin, a campground that was just watered. It's a nice green lawn. It disappeared from all the sites of that elevation except for that nice watered lawn. So campgrounds are good, at least at Belding's. Seems to protect them from local extinction. Oop, there's Tony Lynn doing her thing. So the story with the mammals is there is this general pattern of upward contraction of high elevation species, but very difficult to predict because it varies so much amongst congeners. When we look at life history traits, hibernation traits, physiology, body size, all these ecological variables, we can't predict which species are contracting, which species are expanding, which species are not changing at all. It's hopeless. Now, for birds, it's even more heterogeneous. A whole pile of data here to show we actually have real data. Again, those same three transects. Red is contraction, green is statistically significant expansion. It's a whole mess of greens and blues, reds and greens. Sorry to colourblind men in the audience. And here it's very hard to pick out any overall pattern of change. There is in some places an excess of upwards contraction of high elevation species, but it's very, very heterogeneous. And not what you would expect if all species were responding like robots to this overall pattern of warming of nighttime temperatures. Much more complicated. So, yet there is some sense to it. So if instead of just looking at overall warming across the whole area, if you look at local change in temperature and precipitation, then you can actually explain a lot of the variation. This is what Morgan Tingley called the push and pull of climate change. If you go at a sort of site-by-site basis at the range limits and ask to maintain the same rainfall, given what's changed locally, would you move down or would you move up? Often the direction of predicted movement to maintain the same climate envelope at the limit is downward for precipitation, but upward for temperature change. Right? So that's this push and pull thing. So that's what these arrows are. Red is predicted response for temperature. So this is Yosemite, getting warmer there. You'd expect to move up with temperature change, and down in that same place for if you're tracking precipitation. They're pushing you in different directions. So when we included both of these factors, we could explain a lot more of the species responses than just looking at temperature alone. And I think this is a really important message. We're not the only ones to point it out. Is so much of the focus has been on temperature change, we've really underappreciated the importance of precipitation change, either directly on hydric physiology or indirectly on primary productivity of the systems. And of course, in future modelling of climate change impacts, precipitation is the thing we're least confident about, but it's probably hugely important in predicting species responses. So what are we to do? We've got all this heterogeneity in the system in mammals and birds. There's some common patterns. We are concerned about a number of these high elevation mammals that are contracting, and yet the managers and policymakers want us to come up with simple predictions. Yeah, things are going to move up, this type of species is going to be in trouble, and so on. And it's based on the data, I've got to say it's hard to do. So this is where you have to start the really hard work, the sort of stuff we saw yesterday with the very long-term ecological studies and genetic studies of sort of model systems to try and get a better handle on predicting responses. So thinking about what we can do from the museum perspective, there are a number of things we can extract from the specimens to get a richer view of what's happening. So I've already talked about the top one here, the observed spatial response. We've done a ton of modelling as well, correlative modelling and some physiological modelling. That helps. But the thing I just want to quickly mention is some other things we can do, like looking at changes in phenotypes and genotypes over time, and work that is just starting, led by others in the museum, trying to understand the ecology of what's going on in these systems from early 20th century to now. And of course, all this has to be put in the context of current studies of population dynamics, autoecology, and so on. So our study system here again is these chipmunks. I'm just taking a pair of the chipmunks. They are so cute, these guys. They're really, really lovely animals. And so our poster child for contraction is this alpine chipmunk. It's an endemic to the Sierra Nevada. It's one that's contracting across its whole range. We're really worried about it. And for comparison, we've got the lodgepole chipmunk, which has a broader elevational change, which hasn't changed at all from the historic to the present survey, at least in the elevational range. So we've got a contracting species and a non-contracting species we can compare. So one thing we can do is look at morphology. Right? We've got the specimens, we've got the skulls, body size, body weight and so forth at capture. And the striking thing in this case is that the alpine chipmunk is increasing in body size, or at least skull size, which is our metric of that. And also changing in the skull shape. And this is Ana Paula Assis from Sao Paulo. He's working with Jim Patton, who's really the leader of this work. And they're finding that the rostrum is extending, which he tells me, I'm a lizard guy, not a mammal person, but he tells me that actually has a direct ecological role in retaining moisture, because it expands the nasal membranes and they're better at keeping moisture. So that's something we're seeing in skull shape, and there's a lot more of this sort of work going on now. So maybe from this we can get some clues about what are the challenges the alpine chipmunk is facing and how is it responding ecophenotypically. Now, interestingly, when you look at other species that are contracting, some of the ground squirrels, like the Beldings I already showed you, and also the golden mantle ground squirrel, those two, this is body size on this metric from historical to present, these two also are getting bigger. So the species that are contracting are getting larger in body size, and that's actually independent of elevation. But with warming, under what's his name's rule? Bergman's rule, you expect them to get smaller. What's going on? I don't have a clue. Species that are not changing in range, like beechii, the California ground squirrel, are not changing in body size. So this is a real puzzle. I don't know what it means. Perhaps someone here does. We can also get stable isotopes out of these. So this is sort of historical ecology. You can look at the stable isotopes of whatever, in this case nitrogen and carbon, just from hair samples off the specimens. And these fall into different sort of types of forage for the species as the hair is growing, for the individuals as the hair is growing, and also falling on a precipitation axis in the case of carbon across these different ecotypes. So again, the question is there something happening with diet and so forth? And this is Rachel Walsh's PhD work. This is preliminary data. And the arrows show the vector for a particular location from the carbon isotope and nitrogen isotope signatures from the historic to the present. And in the alpine chipmunk is seeing less nitrogen-15 isotope over nitrogen-14, whereas in the lodgepole chipmunk, which is not changing in range, it's just sort of random noise that's going all over the place. So this may indicate more legume in their diet, and what that means I don't know, but maybe something. The bit I've been more directly involved in is trying to apply genomics to these museum specimens, and this is not necessarily new. People have extracted DNA from museum specimens to look at a small number of genes, the mitochondrial DNA or perhaps microsatellites, for a long time, decades. So again, we have our wonderful draw of chipmunks from 100 years ago, and actually some of these are also the modern specimens from the same place. We were fortunate that Grinnell collected large population series, large population samples, so that we can do population genomics over time and space with this material. So we're interested in two types of questions. We're interested in sort of demographic questions. As species have contracted up the mountains, they become more fragmented in range. Are we seeing loss of genetic diversity through local extinctions? Are we seeing increased fragmentation, genetic fragmentation of populations? We can get at that by looking at the historic to the present population structuring and diversity. And more challenging, but a worthy goal, is can we identify particular genes or pathways that are under strong selection in the declining populations? Because remember, even though population is declining, they're still under selection. In fact, the demographic cost of selection is, in some ways, a cause of the population decline. So can we, given how hard it is to do the ecology, can we help direct the ecologists at the right sort of characteristics or processes by looking at the genomes? I don't know if we can, but we're trying. So to start with, we did Emily Rubidge did the more traditional thing with microsatellites. It was a pain in the neck. Anyone who's done microsatellites on herbarium or museum specimens know how hard this is to do it well. But she persisted, and again we're comparing our non-contracting species to the contracting species. So that the alpine chipmunk, if you project the occupancy models spatially, you can see that in the Grinnell time, they were along the Sierra crest, running along here, but quite continuous in range. Now they're predicted to be much more fragmented as they've moved up the mountains and out of the high elevation meadows. Whereas speciosus hasn't changed in range at all. So the thing that you can see here is pretty obvious, is a decrease in allelic diversity, average number of alleles per locus over time, that's significant, and increased FST, or population divergence. There is no change in heterozygosity. That takes a much stronger bottleneck. But at least drop in allelic diversity, an increase in among population structure, as you would expect given the inferred fragmentation of the range, and no change at all in speciosus. So that was good. That was sort of pretty useful for looking at the demography. What I think is really exciting is this whole skinomics gig. So we've been using exon capture. You can do it with any sort of sequence capture. And the DNA in these skins is perfect for Illumina sequencing. One of the early steps in Illumina sequencing, this massively parallel sequencing, is to shear the DNA up into little tiny pieces. Well, time has done that for us. So that if you look at the DNA out of these specimens, they're often sheared down to 100 base pairs, 150, 200 base pairs. You don't have to do that. You don't have to shear them. You just put it straight into library prep and Illumina sequencing and bang, there's your data. In fact, we get better results off skins than off modern specimens. So this is percent reads on target from an exon capture experiment. So we start with a transcriptome for a chipmunk. We design an exon capture system for, I think, about 12,000 exons in this case. And the orange here are the museum specimens, the blue are the modern specimens, tissues, and these are other species. We get better data, or more data, on target off museum specimens than we get off modern specimens. Now, it's not all routine. There is DNA damage, particularly deamination damage, as you see in any historic work. And the uniformity means we can actually do sort of true population genomic analyses, having controlled for the DNA damage. So this is what we're trying to do. The idea is that recent climate change has caused strong directional selection on pathways related to whatever the ecological challenges are. And so we've got our historic populations, we've got stable species, we've got declining species. So we're expecting to see genomic signatures of selection due to these very recent events. We're only talking maybe 80 generations in the contracting species. So with this amount of data now, having done all the bioinformatics and taken out C to T transitions and so on, which reflect damage, we can look at the allele frequency spectra, which is one of the sort of genomic signatures, and compare that between historical and modern, what's called the two-dimensional site frequency spectrum. So if things are along, all these genes, there's a whole bunch down here and up here, and along the line, that means there's no genetic change from the historic to the present. Ones that are strongly off the diagonal have substantial changes in allele frequency over time. So for example, this one happens to be in a gene to do with oxidative stress, response to oxidative stress, has much higher FST than you expect given the background. Now we're still struggling to do the correct outlier detection tests, taking into account demography, population subdivision and so on. That's ongoing. But I think it looks very promising for being able to identify pathways under selection, which then can help direct the ecologists. Maybe we should pay more attention to oxidative physiology and so forth in our field studies. So I guess the point is that our predictions of the future are incredibly uncertain because we're going into uncharted territory, but we should make much more use of the historic record, and others have said that also at this conference in different ways. Now, this is just a two-point comparison, early 20th century to late 20th century. That's all we have. And this, of course, needs to be combined with long-term ecological studies, as we saw yesterday. But it does help. So the messages are that it's messy. We're seeing a lot of range shifting, but it's very idiosyncratic. It's not all uphill, and even closely related taxa vary considerably in their response for reasons we don't understand. What is clear is that we have to get away from this sort of one-dimensional focus on temperature and think about temperature and precipitation and their interaction along with other forms of habitat change. As was also pointed out yesterday, pollution and other factors. There's strong enthusiasm in the literature, and particularly from policymakers, to apply metrics to predict vulnerability of species to climate change. And I don't think we're even close to being able to do that based on what I've seen in the data. But all is not lost. I think there's a great deal more we can do with Natural History Museum collections, and in particular, and they're in a way they're protected. The data's there, it's protected, we just have to use it. What I'm more concerned about is the loss of long-term ecological data sets. We don't necessarily have the facilities to capture those data in a clean way for future ecologists to work with. Yet, our colleagues who've been doing long-term studies since the 50s, as we saw yesterday, starting with David Lack and others, are retiring, dying, losing their data, whatever. and that's a real tragedy for future ecologists. So thank you very much, and I'm happy to take any questions.